Welcome back, everyone. Today we are going to talk about the central regulation of the cardiovascular system. And when we talk about central regulation, we're really talking about how the brain will affect how our heart functions. We're also going to talk about something called Starling forces. This is different than the Frank-Starling law of the heart. And then we're going to end it with a little discussion on atherosclerosis. Before we get started on the central aspects of cardiovascular physiology, just want to review all of the things that we've talked about so far. I'm going to do that by just putting everything together in an equation. I'm going to share my little whiteboard screen. And so what we've talked about so far, where we ended, was talking about mean arterial pressure being related to cardiac output and the resistance in the arterioles. And so both are direct relationships. If one goes up, the mean arterial pressure goes up. So the one thing I want you to recognize is that some of these formulas, or some of the variables in this formula, can be broken down further. So we don't necessarily just have to talk about cardiac output changing mean arterial pressure. We can break down cardiac output. And if we remember the equation of cardiac output, we know that it depends on the heart rate and the stroke volume. And so now we can look at each feature separately and figure out how it can affect not only the cardiac output, but also the mean arterial pressure. So if we look at the factor heart rate, for example, well, heart rate is going to be influenced by things like the sympathetic nervous system. So SNS input. It's going to be influenced by things like the parasympathetic nervous system, parasympathetic nervous input. If we take sympathetic nervous system, for example, and we say, okay, if we increase sympathetic tone, or increase the input from the sympathetic nervous system, that's going to increase heart rate, because we know from the beta-1 receptors. That is in turn going to increase our cardiac output, and that in turn will increase mean arterial pressure. So just changing one input can have an effect on multiple aspects of the cardiovascular physiology. So I could ask a question about, okay, if you increase sympathetic input, what does that do to the heart rate? You should be able to tell me that it increases the heart rate. I could also ask the same question and say, if you increase sympathetic nervous input, what does that do to the cardiac output? Then you should be able to think about it in two steps. Well, sympathetic input is going to increase heart rate, which is therefore going to increase cardiac output, because our formula for cardiac output is heart rate times the stroke volume. You can have another example. What if we decrease our sympathetic input? Well, if we decrease our sympathetic input, that's going to decrease heart rate. And if we decrease our heart rate, we decrease our cardiac output. If we decrease our cardiac output, we decrease mean arterial pressure. So you can really have a multitude of inputs that you have that will have an effect downstream. When we think about stroke volume, the factors that affect stroke volume, there's a couple of them. If you remember the formula for stroke volume, stroke volume is N diastolic volume minus N systolic volume. And so anything that will either increase end diastolic volume, so increase the amount of filling of the heart or the left ventricle, will increase the stroke volume, or anything that will decrease the end diastolic volume. So anything that will cause there to be less blood in the ventricle after contraction will also increase stroke volume. And so if we think about the factors that affect each of these, there's one major one for end diastolic volume. I know we talked about a few, but they're all related to the same. And when you think increasing end diastolic volume, that's dependent on our venous return. And when we think about something that can change the end systolic volume, that's going to depend on how well the heart contracts. Is it contracting with greater force or less force? So that'll be dependent on the contractility. So let's say, for example, we have, I don't know, we've done something to increase venous return. Say we've given somebody fluids. We've given somebody 2 liters of intravenous fluid. Okay, so this is example A, here's example B, here's example C. We've given intravenous fluid. We'll say we've given 2 liters. Well, that increased volume is going to return to the heart. So that is going to increase our venous return. If we increase our venous return, that means that we're increasing the amount that the left ventricle is filling. So that is going to increase our end diastolic volume. If we increase our end diastolic volume, we are going to increase our stroke volume. That's from the Frank-Starling law that we talked about. That increases stroke volume. If we increase stroke volume, we increase cardiac output. If we increase cardiac output, we increase mean arterial pressure. So that person's blood pressure is going to go up. So you can see that any factor can really have many downstream effects. And then the resistance in the arterioles, we talked about a few factors, one of which was most important. We talked about the length of the vessels. It doesn't change in normal people, doesn't change. We talked about the viscosity of the blood. This is not changed in normal settings. And the biggest factor that we talked about was the radius. This is the most important one. Changing the radius of the arterioles will change the resistance. And the important thing here is that we're talking about radius to the exponent 4. And so it's a very tiny change in radius can affect the resistance to a large degree because of that factor being raised to the exponent 4. So if we give the example of, say, the radius, you have decreased radius of arterioles, that is going to increase the resistance. And that's not only just going to increase the resistance, that's going to increase the resistance significantly because of the variable being raised to the exponent 4. And that increase in resistance is therefore in turn going to increase the blood pressure or the mean arterial pressure. And so when you think about how certain factors can change things within the system, think about these equations. This overall equation of MAP being related to cardiac output and the resistance in the arterioles is really helpful to know. And then you can break it down further. And you can break down cardiac output into heart rate times stroke volume. You can break down stroke volume into end diastolic volume times end systolic volume. And then you can look at how each factor plays in. It's going to take a little bit of time for you to go through this and to really wrap your head around it. But at any point, any example that I give, you don't necessarily have to memorize every example. It's about understanding what's going to happen downstream. And so by understanding the factors that affect each of the variables, you can think through the process and logically come up with the answer. So I just kind of wanted to do that for an overall review of what we've talked about so far. But it'll come up again when we talk about the central control of the cardiovascular system. So we have our central control that that is pretty much dictated by the medulla oblongata, which is in the brainstem. And the medulla has a place called the Cardiovascular Control Center, CVCC. That's the short form for it. And its primary function is really just to maintain blood flow to the brain and to the heart. A lot of different inputs to this cardiovascular control center, but we're going to talk about one main one. And this main one, it picks up signals from pressure receptors that exist within some of the artery walls of our body. These pressure receptors are referred to as baroreceptors. They sense stretch in the arterial wall, and these baroreceptors are located in the aorta and the carotid arteries in the neck. So what they do when they sense stretch, the more stretch they have, the more action potentials they fire to the medulla. And so the more stretch they have, that indicates that the blood pressure is higher. So if blood pressure increases, the stretch increases, and they increase the rate at which they fire action potentials to the medulla. If blood pressure decreases, the stretch is decreased and there's a decreased firing rate of action potentials to the medulla. And so when the signal gets sent to the medulla, the body will respond in a specific way to try to bring the blood pressure back down to a normal value. And we're going to look at the process by which it happens. And it's something called the baroreceptor reflex. So I'm going to give one example of, say, we have high blood pressure. So we have high blood pressure in our arteries, and when that goes across the aortic, within the aorta and the carotid arteries, the baroreceptors there are going to sense the high blood pressure as increased stretch. And that increased stretch in the wall is going to increase the firing rate of action potentials to the medulla. So the medulla is going to get those action potentials, and it's actually going to send out various inputs or outputs to try to bring the blood pressure back down to normal. The first thing it's going to do is it's going to tell the sympathetic nervous system to decrease its activity. And in doing so, that decreased sympathetic activity is going to decrease the amount of norepinephrine that's released, or epinephrine, but it's mostly norepinephrine. They'll both act the same way, in a similar way. So that norepinephrine is going to be decreased less, and that's going to have several effects. Number one, norepinephrine would normally cause vasoconstriction of the vessels because of those alpha receptors, but it's going to cause mostly vasodilation now because there's less effect of it. So it's going to not be able to vasoconstrict, so it'll vasodilate. It's going to decrease the contractility of the heart. So norepinephrine, if you remember from a couple of classes ago, we talked about how norepinephrine acts to increase the force of contraction or increases the contractility. We also mentioned that the other term we used for it was it is an inotropic agent to increase the force. And so that norepinephrine, because there's less of it, it's going to decrease the amount that the force at which the heart will contract. And so we decrease the contractility of the heart. And then the last thing that this norepinephrine will do is there's not going to be as much acting on the sinoatrial node, which is those autorhythmic cells in the right atrium. And we're going to have a decreased firing rate of those sinoatrial nodal cells, and that's going to decrease the heart rate. So the effect of this threefold: vasodilation, decreased contractility, and decreased heart rate. That vasodilation is actually going to reduce the resistance within the arterioles. So it's going to reduce overall resistance in the body, which is a good thing when we're talking about someone who has high blood pressure. We want to reduce that resistance. And that reduced resistance is going to decrease the blood pressure. Because if we remember, mean arterial pressure equals the cardiac output times the resistance in the arterioles. And if you reduce the resistance, it's going to reduce the blood pressure. So we have the effect of reducing blood pressure just from one of those factors. The second thing that the medulla is going to do is it's going to increase the parasympathetic activity. Often when you see one arm of the autonomic nervous system change, the other arm changes in the opposite direction. So decreased sympathetic activity, increased parasympathetic activity. The parasympathetic nervous system acts in one way. You're going to increase acetylcholine that's released, and that is going to act on the autorhythmic cells of the heart. So same sinoatrial nodal cells. More acetylcholine is going to reduce the heart rate. So both the decreased sympathetic activity and decreased norepinephrine, that's going to decrease the heart rate, but so is the more acetylcholine that's released. That's going to decrease the heart rate as well. And these last two factors, the contractility and the decreased heart rate, well, if we look at them, both of those are going to play into reducing the cardiac output. If you're not pumping the heart or squeezing the heart as strongly, you're not going to squeeze out as much volume per beat. So that's going to decrease your stroke volume and decrease your cardiac output. The other factor that affects cardiac output is the heart rate. So you decrease heart rate and you decrease the cardiac output. Both of these together are going to decrease blood pressure. I want to pull your attention to the second last line. The entire point of the baroreceptor reflex is to change the resistance and the cardiac output, both factors that are very important in controlling blood pressure. We don't want to continue doing this until our blood pressure drops to the point that we can't supply all our tissues with oxygenated blood. We want to drop our blood pressure to go back to normal. And there's a negative feedback mechanism. So we have to maintain a good blood pressure. So this reduced blood pressure is going to be sensed by the baroreceptors again, and it's actually going to stop that increased firing rate of those baroreceptors to the medulla. As our blood pressure drops, the firing rate of action potentials goes down to the point where we are going to stop this activity once we reach a normal blood pressure. So it's an example of a negative feedback loop again in our body. So let's have a look at another example of what might happen in the body. So here's the left ventricle. Here's the aorta, the large arteries. We're splitting it now into arterioles. The question is, what would happen if arterial A constricted? It'll actually be the same thing if arterial B constricted or C or D. Just what happens when one of these constricts? Well, if one arteriole constricts, the resistance in that arteriole is going to go up. So as a whole, if we actually look at all of the resistance, the average resistance across all of these, if one of the resistances goes up, on average, the resistance across the whole system is going to increase slightly. And that whole resistance across the entire system is called the total peripheral resistance. So you increase resistance across an arterial bed, you're going to increase your total peripheral resistance. So that's exactly what it does. And if B constricted, you'd have the same thing. If C constricted, you'd have the same thing. If D constricted, you'd have the same thing. If they all constricted, you'd have the same thing. If two of them constricted, you'd have the same thing. And it would be the opposite effect if one of them dilated. If one of them dilated, you'd have decreased resistance. If two of them dilated, you'd have decreased resistance. And so anyways, one constricts, the resistance across that arteriole increases, but across the entire circuit, the resistance is increasing. That increased resistance, we know that that increased resistance is going to affect the blood pressure, because that is a factor that affects blood pressure. Because total peripheral resistance and cardiac output, see that total peripheral resistance is the resistance in the arterioles, times cardiac output is going to affect mean arterial pressure. Increasing that resistance is going to increase mean arterial pressure. That increased mean arterial pressure, that's the increased blood pressure, that's going to be sensed by the baroreceptors. And the baroreceptors are going to fire at an increased rate. They're going to send a signal to the medulla oblongata. And the baroreceptor reflex is going to decrease our cardiac output and decrease our total peripheral resistance back to normal so that our blood pressure goes back to normal. So this baroreceptor reflex, it's exactly the same thing as what we talked about on the previous slides, that flow chart. So we'll talk very briefly about exchange at the capillary level. So once blood reaches the capillaries, there's an exchange of materials that occur across the thin walls. Most cells are within 1.1 millimeter of a capillary. So the diffusion across this very short distance can happen quite readily. The more metabolically active a tissue is, the more capillaries it has, because it requires more surface area for more diffusion to occur so that those cells can get more oxygen and more nutrients. The adult human body has somewhere around 50,000 miles of capillaries, which means that the capillaries, if you line them up end to end in your body, it would wrap around the world twice, which is rather unbelievable, but that's what the textbook says. I don't know who would have done that, or maybe I guess they took a small sample and then just estimated. But the capillaries have a diameter that is barely larger than a red blood cell. And in some cases, the red blood cells kind of have to squeeze through. And this forces red blood cells to sort of pass through in a single file. And when they pass through in a single file, you can get the maximum exchange of nutrients across. We have a couple types of capillaries. We have something called a continuous capillary, which you'd find in your muscles and your nerve cells. The continuous capillaries have endothelial cells that are joined together, but they're somewhat leaky. There's some little gaps between endothelial cells. So here's an endothelial cell, and then there's a second endothelial cell on the right, and you can see that there's a small gap between. And these little junctions between the cells allow for small dissolved solutes like glucose and stuff to pass through. There's also these capillaries called fenestrated capillaries. And if you know the French word for window, fenêtre, it's these capillaries have little windows in them, actually quite large windows at times, and allow for a lot more volume of fluids to pass through. These fenestrated capillaries can be found in the intestines, they can be found in the kidney, and they have fairly large windows that allow for larger volumes of blood or of fluids to pass through. So you can actually see there's a endothelial cell, and between there's all these pores where they're quite large gaps that allow for things to pass through. And so those are found in the intestine because you need a lot of exchange, absorption of fluids, absorption of substances within the GI tract. You want them in the kidney, especially in the glomerulus, where that's the filtration area of the kidney, where the blood needs to be filtered and the fluid component and all the dissolved solutes need to pass through. So if we look at actually the speed at which blood passes through different vessels, we're going to see that the velocity of the blood depends on the total cross-sectional surface area. The aorta gets all the blood, and the speed is quite fast. Anywhere between 30 and 32 centimeters per second, the blood travels. As you get into the arteries, the speed of the blood goes down. In the arterioles, it goes even lower. The velocity of the blood through the capillaries is the slowest. It's only traveling at a couple centimeters per second. And then as you get to the larger vessels, venules, veins, and now the vena cava, you increase the speed again. And so blood moves slower at the level of the capillaries because this will give enough time for nutrients to exchange, so that the time at which those red blood cells are in the capillary is essential to increase or to determine how much diffusion is going to occur. The longer time they spend there, the more diffusion happens. And you can actually see that the lower the cross-sectional surface area, the faster the blood flows. The larger the cross-sectional surface area, the slower blood goes. And so cardiac output is 5 liters per minute. So how can 5 liters per minute flow through the capillaries when the blood flows so slowly there? The answer to that is that you have all 5 liters of blood going through a very short distance, but there's so many capillaries that you only need to travel that short distance. You're not traveling you're not having all the blood going through a single file of capillaries. That would take, that's 50,000 miles of capillaries. You literally have billions and billions of capillaries, and each one has a little bit of blood that's going through slowly, and collectively, you're going to end up with 5 liters of blood per minute over the entire cross-section of capillaries. So the largest cross-sectional surface area has the lowest velocity. So collectively, all the capillaries in the body together have a flow rate of 5 liters per minute, even though in an individual capillary, blood's traveling quite slowly, but collectively it's 5 liters per minute across all capillaries. So capillary exchange happens by a couple of processes. There's diffusion, which is a passive movement of substances from an area of high concentration to an area of low concentration. And so this will be how oxygen and carbon dioxide are going to move across. There's also the process of transcytosis. And this is for movement of larger proteins, larger solutes that end up getting transported across via these little vesicles that travel across the cell membrane. And so some of them actually travel right across. Some of these vesicles will fuse and create temporary channels so that the solutes can pass through. The one concept that's extremely important to understand for this section, and it'll come back when we talk about the kidney, is the idea of bulk flow. And this is the massive movement of fluid between the capillaries and the space between the cells. And so the movement of fluid, there's a couple of definitions. Movement of fluid into the capillary, so from the interstitial space and into the capillary, is referred to as absorption. And movement of fluid, if it's leaving the capillary and going into the interstitial space, that's referred to as filtration. There's a couple of terms there's a couple of pressures that you really have to know. The two pressures that really regulate this bulk flow and determine whether fluid's going to leave the capillary or fluid's going to come into the capillary are the first concept is hydrostatic pressure, hydro meaning water. And so this is the pressure that's created by the fluid itself. So if you think about fluid inside of a vessel, like a capillary, that fluid is going to push on the walls and create a certain amount of pressure. That pressure from the fluid itself is referred to as the hydrostatic pressure. The second pressure that's important is this pressure called colloid osmotic pressure. And it's sometimes also referred to as oncotic pressure. And this particular pressure, it's denoted with the symbol pi. Hydrostatic pressure is denoted with the symbol P. This pressure is actually created by proteins itself that are within the fluid space. So if you think about your blood, it has a lot of proteins in it, especially in the plasma. And those proteins are actually going to work to draw water in, almost like osmosis, like it's going to try to dilute those proteins and it's going to pull the water back in. That's what the colloid osmotic pressure does. So you can imagine the pressure of the fluid inside of the vessel is trying to push the fluid out, but the presence of those proteins within the vessel is trying to draw fluid back in. And so it depends. Is the pressure trying to push the fluid out? Is it greater than the pressure trying to pull the fluid back in? Or is the pressure of the proteins trying to pull the fluid back in greater than the pressure trying to push the fluid out? And so the balance of these forces is going to tell you whether you're going to have a net flow of fluid out of the capillary or a net flow of fluid going into the capillary. Together, these forces are referred to as Starling forces. It's the exact same person who did Frank Starling law, but Frank was not part of it. So it's the exact same guy, Starling. So they're called Starling forces. So the colloid osmotic pressure, so again, sometimes referred to as oncotic pressure, is much higher in the plasma, so in the fluid component of blood, than it is in the interstitial space. And the thing to really know is that for our purposes, the amount of protein that exists outside of the capillary and in the space between the cells, for our purpose, is essentially zero. There should not be any protein in that fluid, in the interstitial fluid. There should be zero protein. So the amount of pressure that exists in the interstitial space is zero. There's no pressure in that area. Compared to the pressure, the colloid osmotic pressure that is within the capillary, there's lots of protein in the blood. And so that the plasma that's going through the vessel has quite a bit of protein. It's trying to pull in fluid. It has quite a bit of osmotic pressure. And so the pressure, the colloid osmotic pressure of the plasma is 25 millimeters of mercury. It has lots of protein. And the thing to note about this is that as you go from one end of the capillary to the other end of the capillary, this colloid osmotic pressure doesn't change at all. It stays 25 within the plasma through the duration of the capillary. I'm going to show a picture in a second that'll help put this all together. The hydrostatic pressure, so that pressure that's pushing on the walls of the capillary, the hydrostatic pressure, it does change across the length of the capillary. At the arterial end, there is a higher pressure pushing of the fluid pushing on the wall. So the arterial end of the capillary has a hydrostatic pressure of somewhere around 32 millimeters of mercury. And the venous end of the capillary has a hydrostatic pressure of somewhere around 15 millimeters of mercury. If we look at the hydrostatic pressure of the interstitial space, the fluid that's between the cells itself, it's very low. For our purposes, we're also going to call that 0. So the hydrostatic pressure of the interstitial space is 0. And so if we take the hydrostatic pressure, that pressure trying to push the fluid out, and we subtract the colloid osmotic pressure trying to pull the fluid back in, that's going to give us the net pressure. Are we going to push fluid out, or are we going to pull fluid back in? If this gives you a positive number, hydrostatic pressure minus colloid osmotic pressure, that's where you take hydrostatic minus colloid osmotic. If that gives you a positive number, there's going to be a net filtration. So there's going to be a net pressure pushing the fluid out, then pulling the fluid back in. So that we call that net filtration. If we get a negative number, that's going to mean that the colloid osmotic pressure is higher. And so the pressure pulling fluid back into the vessel is higher. And so our net effect is that we are going to overall pull fluid back in to the capillary. So let's look at a picture here. We're going to put all the pressures that we talked about. This is a capillary, and the fluid within the capillary, this is the arterial end of the capillary, and this is the venous end of the capillary. Remember I said we're going to first look at the osmotic pressure within the capillary, those proteins that are in there. And remember we said that osmotic pressure does not change from one end of the capillary to the next. So we have, at the arterial end, we have an osmotic pressure of 25 millimeters of mercury. Along the length, it doesn't change. So at the venous end, we expect to have the exact same. So we have a plasma osmotic pressure of 25 millimeters of mercury. The hydrostatic pressure, though, that pressure pushing the fluid, pushing on the walls of the capillary, at the arterial end, we said that hydrostatic pressure was 32 millimeters of mercury. And we said that hydrostatic pressure decreases across the length of the capillary. And so our hydrostatic pressure at the venous end is lower, 15 millimeters of mercury. So if we're looking at the arterial end and we subtract the two values, we see that the pressure of the fluid pushing on the walls, trying to push the fluid out, is higher than the pressure of the proteins trying to pull the fluid back in. And so we get a net pressure of 32 millimeters of mercury minus 25 millimeters of mercury, which is overall positive 7. And when there's a positive net pressure, that is a net filtration pressure. So we're pushing fluid out, and we get net filtration at the arterial end. At the venous end, if we look at the difference between those pressures, we have 15 millimeters of mercury minus 25 millimeters of mercury. That's going to give us a net pressure of negative 10. And therefore, at the venous end, we're actually going to have a net pressure trying to pull fluid back in, or a net absorption pressure. So there's net absorption at the venous end. So you're pushing fluid out, getting exchange of things, and then pulling fluid right back in. That's really what's happening at the level of the capillaries. You may need to go through that a couple of times to really make it make sense. There is I think I'm going to be talking about, yeah, so this bulk flow, after all the filtration has occurred and all the absorption has occurred, this bulk flow doesn't actually regulate our fluids very well, because when all is said and done, there's still about 3 liters of fluid that is left in the interstitial space that your capillary bulk flow doesn't pick up. And so if you think about it, per day, if 3 liters of fluid is leaving your capillaries, that's almost your entire blood plasma volume. So the question is, how do you get that extra 3 liters back into your blood vessels? And the answer to that is the lymphatic system. Most people think of the lymphatic system as part of the immune system, which is true. Our lymph nodes are part of the immune system and we have immune cells. But the lymphatic system is an important collection of vessels in the body that allows for that excess interstitial fluid that can't get absorbed into the capillaries after bulk flow. It gets it back into the blood vessels. And so that 3 liters is going to go back in, is going to enter the lymph. The lymph is going to help you out, and then it'll eventually be returned to the blood. So you can imagine that if you have a blockage in your lymph vessels, it cannot reabsorb all that fluid, and that fluid in the area where you have a blockage will just keep accumulating and accumulating and accumulating. And someone might end up with swelling in that area of their body where their lymph vessels aren't working well. And that swelling from fluid is referred to as edema. That's just a fluid accumulation in the interstitial space. So there's actually a lot of things that can lead to edema and lead to fluid entering the interstitial space. So one common thing that can lead to edema is if you have a situation where the hydrostatic pressure in your capillaries increases. And a common reason why the pressure of the fluid can increase is that you have more fluid in the capillaries. And so one of the reasons is heart failure. If someone has heart failure, it means their heart can't pump the blood that's sent to it effectively enough. And if you imagine, that blood is going to back up through the circuit. So if we're talking about the right side of the heart, for example, we're having blood going from the large veins in the vena cava into the right atrium, into the right ventricle. If that right ventricle can't pump properly, you're going to have blood back up into the right atrium. And blood is going to back up into the vena cava. And blood's going to back up into the large veins. And blood is going to back up into the venules. And then it's eventually going to back up into the capillaries. And that increased fluid in the capillaries is going to push to a greater extent on the walls of the capillaries. And that fluid, that extra fluid, is going to be pushed out into the interstitial space. So a person who has an increased hydrostatic pressure is going to have a lot more filtration of that. So that bulk flow is going to be net filtration across the entire length of the capillary, and you're going to have more fluid entering the interstitial space. And so it's going to lead to edema. Another thing that can lead to edema is if you have a decreased amount of proteins in your plasma. So there's less proteins that are trying to pull the fluid back in. So a common reason that someone might have decreased proteins in their blood is that their liver isn't working. Liver is a really important place where you make a lot of your plasma proteins. So people in liver failure don't make those plasma proteins. So if you think about how much protein is in someone with liver failures passing through the capillaries, it's a lot less than someone who has a normally functioning liver. That lower amount of protein is going to reduce the plasma osmotic pressure. And there's going to be less ability to pull the fluid back into the vessel. And so with less ability to pull the fluid from the interstitial space back into the capillary, you have less absorption. And overall, you have more filtration, and more fluid is going to get left in the interstitial space. And that's going to present like edema. So there's a lot of things that can cause edema, and it's about trying to hone in on the exact cause. I won't expect you to hone in on the exact cause if I give you a situation, but I might tell you about something going on. I might say, oh, someone has liver failure. They don't make as many proteins in their blood. What is a plausible physiologic explanation for why they may have edema in their, I don't know, in their ankles? And so then you'd have to think, okay, well, they have less protein in their blood, so less plasma osmotic pressure. And with less plasma osmotic pressure, you're going to get less absorption of that fluid. So it's absolutely essential for you to really understand the bulk flow, because that will tell you what direction fluids are moving. So make sure you go through that a couple of times. It might take 2, maybe 3 times for you to really read through it and really understand the concept of the direction that things are going. Is it a filtration thing or is it an absorption thing? I'm going to draw out one image that might help you a little bit. I'm going to draw this on the whiteboard area. Where is the whiteboard area? There it is. Share that. Okay, so I'm going to get to a new diagram. And so one thing that might help you understand this is, so if we're looking at the pressure, this is going to be pressure in the capillary. So this is millimeters of mercury. And this is across the length of the capillary. So we're going from the arterial end to the venous end. So this might help you understand the graphical representation. So if we think about I'm trying to think of what color would be best to do this in. We're gonna go with purple. Okay, we're gonna go with the purple. Purple. Oh no, I don't want that. Okay, purple. Okay, good. I think I have purple. No, I don't. Okay, we're not doing purple. I can't figure out how to do that. So we're just going to go with we'll go with black and green. Okay, so as we mentioned, from the arterial end to the venous end, our hydrostatic pressure goes down. It starts high and it goes down. So we started at, I think it was 32 millimeters of mercury, and we went to 15 millimeters of mercury. 15 millimeters of mercury. That's what the textbook talked about. And then our plasma osmotic pressure, I apologize for using green because arterial's in red, and if anyone has red-green color blindness, I apologize for that. So this, I better label this, that this is the plasma, this is the plasma osmotic pressure, or this is the hydrostatic pressure. This black line is hydrostatic pressure, and that's within the capillary. And then our green line is going to be our 25 millimeters of mercury. We'll put it somewhere around, I don't know, here. And we said that doesn't change across the length of the capillary. So it is absolutely staying the exact same. And so this is our pi. And this is in the capillary. And this is somewhere around 25 millimeters of mercury. Doesn't change at all. Our hydrostatic pressure goes from 32 to 15. Well, this graph actually essentially tells you whether you're having a net filtration or whether you're having a net absorption. So if we look at this end of the graph right until they cross, we can see that at every single point, hydrostatic pressure is higher than the oncotic pressure or the plasma-osmotic pressure or the colloid osmotic pressure. It's higher in every instance. So across this area, this entire area is a net filtration, because your pressure pushing fluid out is always higher than your pressure pulling fluid back in. So this is a net filtration. After these two lines cross, you can see that your plasma osmotic pressure is always higher than your hydrostatic pressure. So at some point in time, near the venous end, you are getting a net. I'm going to try to open up orange again. You're going to get a net absorption, because your oncotic pressure is always higher than your hydrostatic pressure. This is net absorption. Hopefully that helps it make sense a little bit more, and helps it put it into a graphical representation. So this area, you can see that your oncotic pressure is always higher than your hydrostatic pressure in this second part of the graph. In the first part of the graph, you can see that your oncotic pressure is always higher. Your hydrostatic pressure is always higher than your oncotic pressure. And so that's always at the arterial end compared to the venous end. Hopefully that makes a little bit more sense. And then you can sort of work through some other examples. I'll actually put one of the examples down to show you that. So we're actually going to draw out the exact same thing. We have the arterial end versus the venous end. Let's look at the example of, we're going to look at the example of decreased osmotic pressure. Remember, we said that could lead to edema because you're going to get more filtration. Decreased osmotic pressure for something like liver failure. Okay, well, we have our 32 millimeters of mercury, our 15 millimeters of mercury, 32 millimeters of mercury, 15 millimeters of mercury. That's where our hydrostatic pressure kind of goes from. It goes from 32 at the arterial end to 15 at the venous end. But in the case of liver failure, well, in our normal setting, we had an oncotic pressure of 25 millimeters of mercury. Let's say that this liver failure is going to cause there to be an osmotic pressure, even if it goes down somewhat. Our oncotic pressure, let's say that it's, I don't know, it goes down to 18 millimeters of mercury. Well, that's not going to change. As we know, our oncotic pressure doesn't change along the length of our capillary. From the arterial end to the venous end, we have the exact same oncotic pressure. In the case of liver failure, we might have this oncotic pressure of, I don't know, I'm just making it up. So 18 millimeters of mercury, quite a bit less than 25. And let's look at the part of the curve that is now doing filtration. We have a huge area that's doing filtration now, comparatively speaking. There's much more filtration, much more fluid getting pushed out of the capillary. We have more filtration and very little, very little absorption compared to the image above. And that higher amount of filtration, that's going to gets put into the interstitial space, and that leads to edema. That's swelling because you have more fluid getting pushed there. So hopefully that also helps you see the relationship between more filtration, more fluid's going to get pushed there. I mean, we can go through other examples, but that's the gist of it. That is the gist of it. So hopefully that makes sense to you. We're going to go back to our slide deck. But I encourage you to draw out an example of heart failure where your hydrostatic pressure is going to be changed to the point where, well, instead of 32, it's actually going to increase. And I want you to figure out what's happening when your hydrostatic pressure increases. And so draw out the graph and see whether you can see that, yes, there's going to be more filtration, and see if you can work it out. And if you're having difficulty, let me know, and I'll go through I can go through another example maybe during the review session before our next midterm. Okay, we're going to share the diagram here. So those are things that can lead to edema. We're going to talk very briefly about atherosclerosis, and then we're going to end our cardiovascular section of this course, and we will then move on to our section on blood, and then very nicely transition into our respiratory section. So a little bit of atherosclerosis. What is it? It's really narrowed arteries, and it can be narrowed arteries anywhere. We're going to talk a little bit about how it occurs and then why it's such a bad thing. And so here's how atherosclerosis occurs. So here's a normal arterial wall. We have a lining of endothelial cells, those flat endothelial cells of a blood vessel. Underneath of that, we have some elastic connective tissue in an artery. Underneath that, we have some smooth muscle that is going to help with, control the diameter of that vessel. And then we have some fibrous connective tissue on the outside. So this is what happens in normal circumstances. Well, the first thing that happens to develop atherosclerosis is that you end up with something called a fatty streak, which is really just that bad cholesterol, that LDL cholesterol, that accumulates between the endothelial lining and the connective tissue. It gets placed there and it accumulates. So you can see that yellow area, that's all our LDL cholesterol. And while it's there, it actually gets oxidized in that area. So it gets modified slightly and it turns into a fatty streak. As things develop, there are immune cells within this area that will start to ingest this LDL cholesterol. And the funny thing about these immune cells, they're macrophages, so they ingest the cholesterol, but they don't have an off switch. They just keep eating it and eating it and eating it and eating it. And because it's sort of a lipid-containing molecule, that lipid-containing molecule will be seen, if you look at those macrophages under the microscope, they look like there are all these little droplets of lipid within them. And they kind of look like there's foam within them. So they call them foam cells. And eventually those macrophages get larger and larger and larger and larger. They just don't have an off switch to stop eating the LDL cholesterol. At the same time, smooth muscle cells, which are underneath, get attracted to the macrophage because the macrophage is releasing these inflammatory mediators. They're saying, okay, there's something invading us. We need to recruit more cells. And so the smooth muscle cells are recruited there into this area just under the endothelium, and they start to divide. They also start to take up cholesterol. And eventually what happens is that you have so much stuff that is accumulating underneath of the endothelial lining that you get this growth on the inner surface of the blood vessel that is full of lipid and full of all of this cholesterol and full of these cells, and it pushes into the lumen of the blood vessel. So you can imagine that if this was a whole blood vessel, the luminal diameter is starting to narrow now. At this stage, it's called a stable fibrous plaque. So there's quite a bit of stability to this. It doesn't tend to break open very easily because it's fairly stable at this stage. All the inflammation causes there to be some fibrous tissue that's laid down, and it lays down tissue that's almost like a scar formation to try to wall off this lipid core that's happening underneath of the blood vessel. Well, eventually what's going to happen is you're going to get some calcium deposits in there. So this area starts to get hardened with the calcium deposits, and that's the result of the inflammatory process that's going on in this lipid core. But eventually what happens is that those macrophages start releasing so many inflammatory cytokines and so many inflammatory mediators that some of those inflammatory mediators start to digest the fibrous tissue and starts to make this very stable plaque. It starts to turn it into something that can become very easily ruptured. And that's where we have problems. And this is called a vulnerable plaque. This can happen in any artery, but there are places where it tends to happen more frequently. This vulnerable plaque is now no longer stable, and it's vulnerable to rupture. And so when it's vulnerable to rupture, this is where problems occur. When the endothelial cells, there's a break in them, and the collagen that is underneath of here from the scar formation and from the connective tissue underneath, when that collagen is exposed, that's where we have a problem. And so why is this a problem? So we're going to talk about the physics around all this. So the first thing that I want you to make note of, and so this is a coronary vessel. We're going to talk about the blood vessels around the heart. So the coronary vessels. And this is blood flowing through the coronary heart. And yes, it's in green. It's kind of close to Halloween. So green blood, sure. And we're just going to say, for our purposes, that the radius is one something, whether it's, I don't know, it's definitely not one, but we're just going to say radius 1 for our purposes. Normally, in a normal coronary vessel, the blood flow follows this very beautiful parabolic motion. And this flow is called laminar flow. And it follows this very nice parabolic path. It very smoothly flows through the coronary vessels, nicely gives oxygen to all the cardiac muscle cells. And while we're doing this, the flow through this artery is very much dependent on that Poiseuille's law. So where flow and radius are directly related to each other. The higher the radius, the better the flow. Higher the radius, the better the flow. This is very similar to saying, well, the lower the radius, the higher the resistance to flow. So the lower the flow. But we're saying that flow is dependent on radius. Higher the radius, the higher the flow. And it's radius to the exponent 4, just like we talked about before. So now the question is, what would happen if we reduce the radius by 2. It's not really by half. If we reduce the radius by half, then we're just going to cut the radius in half. Well, what's going to happen? We now are putting this plaque in here, it's atherosclerotic plaque, and we're cutting the radius in half. Well, here's what's happening now. The flow across this vessel is now going to be reduced. But because it's a relationship of r to the fourth, if that radius turns out to be 1/2 to the exponent 4 is 1/16. So we actually reduce the flow by a factor of 16 times. So this is going to be a number that is a bit ridiculous, but if we have 1 liter of blood flowing across, if we reduce the radius, cut it in half, we're going to have 1/16 of that blood flowing across. So much less flow going across that area. The problem is, in order to get the same flow across that area, we really got to increase the pressure of that blood flowing through there. And that blood, the heart needs blood, so the pressure needs to increase. And the pressure needs to increase also by a factor of 16 in order to go across. And that high pressure, as you go through that plaque, becomes quite dangerous. It gets rid of that very nice laminar blood flow as it gets to that plaque region, and it turns it into something called turbulent flow, whereby instead of beautiful, long, flowing blood, now the blood starts to flow in this haphazard arrangement and starts to hit the walls of the blood vessel, starts to hit the plaque at quite a high pressure. And that high pressure across that plaque can rupture it. So now we have a ruptured plaque, which is very bad. Because the ruptured plaque, that's going to expose collagen. And as blood flows through there, the platelets in your blood are going to sense, oh, there's collagen here. There's an injury to the body. And those platelets are going to start to stick. They're going to try to fix the damage to that blood vessel wall. They think they're helping, but they're not, because those platelets are going to stick and they're going to aggregate together and they're going to completely block off this vessel. And one thing about the heart is that every part of the heart only has one blood supply. So if you cut off this blood supply, the heart muscle downstream from it completely dies. And this would be called a heart attack. So somebody would be having a heart attack. The tissue after this ruptured plaque is going to die. And so one of the things that we can do in this situation, I'm not going to get into the ins and outs of treatment of heart attacks, but one of the things that can be done is, for someone who is having a heart attack, is that they're told to chew aspirin. And yes, aspirin is an anti-inflammatory medication, but it also is a medication that inhibits the ability of platelets to stick together. And so people are told when they're having a heart attack to chew a couple of baby aspirin, those 81 milligram aspirins, and to prevent more platelets from actually aggregating. But that's really the physics of atherosclerosis. We're not going to get into more than that of it, but it's highly dependent on that radius to the fourth and the decreased flow of blood going to an area of the heart. So that Poiseuille's law is exceptionally important. So I'd encourage you to go through all of the equations that we talked about, but less so to necessarily calculate. You might have to calculate stroke volume here and there, or cardiac output here and there, but it's more so putting together all the equations so that you can understand how one factor can change any other factor and what the overall effect will be. I also want you to be able to go through and understand the concept of bulk flow, so that when if you were told hydrostatic pressure increases, or the level of proteins in the blood decreases, or the level of proteins in the blood increases, I want you to be able to predict what is going to happen at the level of the capillary based on the changing hydrostatic pressure and the changing colloid osmotic pressure. And so really go through different examples and try to figure out the general trend of what's going on. So we'll end it there today, and I will see you next day. We'll start talking about our section on blood. Have a great day, everyone.